Could CRISPR be obsolete? A new technology called bridgeRNA, reported last month in Nature, could be the one to finally take CRISPR down from the top of the hill.
Since its inception in 2010, CRISPR has been the easiest, cheapest, and most efficient way to perform genome editing, taking over technologies like zinc fingers and TALENs.
However, as we have mentioned many times in our Making the Cut series, one of the main limitations of CRISPR-mediated approaches is the need to introduce breaks in the DNA, which can lead to unwanted outcomes.
In more recent episodes on Making the Cut, we told you about how technologies like base editing, prime editing, and click editing can be used to increase editing efficiencies while lowering DNA double-strand breaks almost to zero.
However, some concerns still exist because while these technologies do reduce double-strand breaks, they don't completely eliminate them.
In contrast, the new system we will talk about today, bridgeRNA, bypasses all these issues by not using Cas9 at all. The key once more are recombinases.
Now you must be thinking that we'll be talking a lot about recombinases later, and you'd be right.
Indeed, in our last Making the Cut episode, we talked about combining prime editing and recombinases to insert large cargos, and our last video showed the evolution of the system called EEPAS.
However, in both these cases, while the recombinase was doing the heavy lifting of the integration, prime editing was still required to insert the target sites for the recombination to happen.
Normally, recombinases can only recognize very specific sequences which have to be added to the target gene for them to work. But what if we could use programmable recombinases?
This is where a new publication from the lab of Patrick Hsu at the Arc Institute comes in.
They investigated mobile genetic elements that are able to insert and excise themselves from our DNA and discovered that some of them use recombinases that can be programmed through RNA.
The same way that Cas9 binds to a guide RNA, this recombinase also binds to an RNA molecule. However, the cool thing is that this RNA molecule has two recognition sites that can bind to two different DNA sequences.
That allows the recombinase to bring together those two sequences and make them recombine. For this reason, they call this RNA molecule bridgeRNA.
This bridgeRNA molecule has a pretty complicated 3D structure which we are not going to get into. But what you need to know is that there are two main loops which recognize the two DNA sequences.
And differently from the CRISPR-Cas9 system, where each guide RNA recognizes only one DNA strand, the bridgeRNA recognizes both DNA strands at the same time, which makes its design a bit more complex.
However, the authors showed that this system is programmable, and changing the recognition sequences in the two loops can direct the bridgeRNA recombinase complex to different DNA sequences.
And this can be used in a cell DNA to produce excision or inversions, but it can also be coupled with a donor to perform targeted integrations.
And all of this without cutting the DNA at all, which promises a very high safety profile, and with pretty good efficiency.
However, the system is far from perfect. Off-targets are still a thing, and a better characterization is still necessary.
We expect that the system will undergo a similar evolution to what CRISPR went through in the first years, where variants with higher efficiency and specificity will be developed soon.
However, adding this shiny tool to our gene editing toolbox will expand the possibilities when designing a novel gene editing approach.
So you can breathe out, CRISPR is still king of the hill, but the bridgeRNA system promises fierce competition in the years to come. We here at CRISPR will follow this new technology closely and tell you all about it.